this is Mrs. Unland. Let's give this a try. I'm going to attempt to record myself talking about this PowerPoint and drawing in things so that you can copy everything into your notes. So the first few slides are just going to be pictures that you do not need to copy, but I will let you know when you need to start writing things down. So today we're going to talk about gene expression and protein synthesis, which basically we're getting into talking about how does your body take this strand of DNA that we know has genes on it and convert that into the way that your body is built during fetal development and the way that your body functions after that point. So what happens is that each gene on your DNA is responsible for building proteins, and depending on the order of the A, C's, T's, and G's, it builds proteins that have different shapes and different functions. So some proteins might be responsible for breaking down sugars found in milk. You have myosin, which you should have learned a little bit about in Bio 20 for muscle contraction, collagen, hemoglobin, etc. Every gene builds a different protein, and proteins function based on their shapes. So we know that every cell in our body has all of the information that we need. Every cell has all 46 chromosomes, every cell has all the DNA. A nerve cell needs to do different things than a muscle cell, and that needs to do different things than an intestinal cell. So what happens is that sometimes genes can be turned on or off, and that was part of what you talked about in the notes for the biotechnologies the other day, where in humans we put those methylase, or we use the methylase enzyme to put a methyl group on to physically block a gene from being able to be read. So when we look at things like a nerve cell, some genes will be turned on, some genes will be turned off, and those will be different genes than the ones that are turned on or off in, for example, a muscle cell. We also know that not everything about the way that our body works is completely controlled by genetics. There's other factors like diet, exercise, social interactions, toxins, etc. And most things are a combination of both. From the video the other day with the personal DNA testing, the guy was shown that he had a certain % chance of getting different diseases, which means that he has a genetic influence, but then things like his diet, exercise, etc. will change his likelihood of having a heart attack, getting cancer, etc. So we're going to talk about how your body reads the DNA to make a protein to then go out into the rest of the body and tell it how it should function. Okay, so here's where you need to start writing things down, talking about protein synthesis. So this is the way that information in a gene is converted into a specific characteristic or trait. Okay, and we call it gene expression. So I'm going to draw a diagram. You need to copy this diagram down as well. Ms. Atkinson can pause the video at any point if you guys need some more time to write stuff down. I'm just going to continuously talk. So the diagram should look like this. You have a long strand of DNA, and then within that DNA we have different sections where we have genes. So I'm just going to color in a couple little chunks with red. This is representing the genes. In front of every gene there is another section called the promoter. Okay, so there's an area in front of each gene called the promoter. There's also an area behind each gene called the terminator. And this is going to come up again. So every gene has a terminator at the back and a promoter sequence in front of it, and we'll talk in detail about what that means as well. Okay, so I'll just give you a second to copy that diagram down. Again, certain areas of the body will be using certain genes. Okay. Next slide. What is a protein? A protein is just a long chain of amino acids. You should have talked about that a little bit in Bio 20 when you did your digestion unit. Every protein that your body makes is somewhere coded for in the DNA. And again, in Bio 20 you should have talked a little bit about how the function of a protein is completely dependent on its shape. So you talked about how it has primary, secondary, tertiary, and quaternary folding to give it a certain shape. And its shape controls what it does. So depending on where it is, it might be responsible for muscle contraction. All of our enzymes that the body makes are proteins. A lot of the hormones that we talked about earlier are protein hormones as opposed to steroid hormones. We know that in the cell membrane we have protein channels, or things like our sodium-potassium pump. And the diagrams that you can see at the bottom of the screen here, whoops, sorry, I'm just trying to control my mouse with my left hand. Okay, the diagrams down here, so these are just showing the different shapes of some different proteins. So the central dogma, Mrs. Atkinson can probably tell you a little bit more what that means. It's kind of a social studies term, but basically that means that this is the big idea. This is the big idea of DNA. So there's two main stages, transcription and translation. So if I have DNA and I convert it into something called RNA, we'll talk about this, and then I can also then convert that RNA into protein. I hope you can see this blue pen okay. Maybe I'll grab a different color. We know that DNA replicates itself during interphase of mitosis. The process of going from DNA into RNA is called transcription. So you can write transcription under this arrow here, and the process of going from RNA into a protein is called translation. You guys need to know those names, and you need to know what they're changing. So DNA into RNA is transcription, RNA into protein is translation. Transcription happens in the nucleus. We know that because that's where DNA is housed. And then translation actually happens in the cytoplasm. So we'll get into those details as we get to that process a little bit more. So you need to have that whole diagram, please, copied down. The diagram below is the one out of your textbook. So again, it's just showing DNA in the nucleus. The process of transcription converts it into mRNA. It leaves the nuclear envelope, and then translation will convert that into a polypeptide, which is just a large peptide chain, which is what a protein is once it then folds in on itself. Okay, just have to delete this. There we go. And on to the next slide. Okay, so what is RNA? I talked about this thing that DNA gets converted into RNA. So RNA is ribonucleic acid. If you remember, DNA is deoxyribonucleic acid. Similar, but a different sugar. So it's a ribose sugar instead of a deoxyribose sugar. The other difference between RNA and DNA is that RNA is single-stranded. Okay, so the nucleotides do not pair up. And then the last thing is that instead of having thymine as one of our nitrogen bases, we have uracil. So A pairs with U instead of T, but guanine and cytosine still pair up together. There's two different types of RNA that are found. The first is called mRNA, and the M stands for messenger. Okay, this is what's responsible for transferring the genetic code out of the nucleus and into the cytoplasm. We just talked about that it is single-stranded. tRNA, the T stands for transfer. Partially the way you can remember this too is that it actually kind of looks like a T shape. So you can see this diagram I'm circling down at the bottom of the screen. That is what tRNA looks like. It's responsible for carrying specific amino acids to the ribosomes. Ribosomes are where proteins are made. And there are 20 different tRNA molecules, each which corresponds to one of the 20 different amino acids. So that little AA abbreviation stands for amino acids. And then one other just sort of definition here that you need to be familiar with is what is a codon. So when we talk about an mRNA codon, we're talking about a triplet of nucleic acids, okay, or 3 nucleotides, 3 nitrogen bases. So DNA is always read in a group of 3 of the nitrogen bases at a time. Whoops, sorry, okay. No, so now we're going to go into the process. So the process of transcription to start with, there's 3 different steps. Transcription begins with initiation. Okay, this deals with that promoter sequence that I drew in that first diagram. So initiation, you have the RNA polymerase, which is an enzyme that you need to know, binds to DNA and it scans along until it finds a promoter. And the promoter is just going to be a sequence of adenines and thymines in any order, but the key thing here is that there's no cytosine or guanine interspersed anywhere throughout it. Okay, so it's just attaches to the DNA, scans along until it finds a sequence of A's and T's, which is the promoter. Okay, the next step is elongation. So once it finds that promoter, it separates the two strands of DNA. You can see that you have the one strand open at the top, and it's going to start to build a single-stranded RNA. Again, direction is important, just like in DNA replication, has to be in the 5' to 3' direction. So it's only going to be using one of the strands. The strand that it uses is called the template strand, and it's just going to scan along, pairing up the nucleotides, but again, this time we're going to be using uracil in the place of thymine. Okay, so I'm going to draw in a diagram, and I am going to make this clear. Okay, just give me 1 second here. There we go. Okay, so I'm going to draw in a diagram and you should copy this down. So if this is my original strand of DNA, comes as a double-stranded RNA polymerase separates apart the two strands. So I'm going to draw in a sequence here and you need to copy this down. Okay, and then if this is my strand below, it should have the complementary base pairings. So A with T, C with G. Okay. This was a double helical strand of DNA. When RNA polymerase attaches to it, it opens it up. Okay, so if this is my template strand, you'll need to be told which strand is your template strand. So I am telling you that we're going to use the bottom strand as a template strand. RNA polymerase is going to come in and build an RNA strand using that as a guideline. So if I have a T, it's going to pair up with an A. Okay, and it's just going to read down in the 5' to 3' direction, building a strand that pairs up. Okay, big thing that's different here though, A normally pairs with T, but in RNA there is no T, so it pairs with a U instead. Okay, so I have now this red strand, which is my mRNA, which has been built using one of the exposed pieces of DNA as a template. Okay, I'm going to erase this, so pause if you need some more time to finish writing it down. Okay, the last step, so we had initiation, elongation, and then the final step is termination. So this is when we are finished with the sequence. So the RNA polymerase detaches, the DNA strand goes back together, and you're left with this single strand of mRNA, which is shown in this picture here as the green. And it now is able to leave the nucleus. It's much smaller, it's only single-stranded, so it can actually fit out through some of those holes in the nucleus, which we call nuclear pores, and get out into the cytoplasm. Okay, so what you end up left with, I'm just going to get a new slide here again, make it blank and white so that you can see what I'm drawing. I'm left with now this strand, so this is the same strand that you had originally, which is in the sequence AA, C, G, A, U, okay, and it is able to now leave the nucleus and go out into the cytoplasm. I'm going to delete this and go to the next slide. Okay, so here is what I would like you to do. Again, you can just stop the video, Go to your textbook and try the practice questions on page 669. Numbers 1 and 2 will have you simulating the process of transcription.